So when I start talking to audiences, not usually as experienced as this one and as diverse, I usually start with some key questions. So what if there were no new antibiotics? So we should obviously use the ones we've got really wisely, and many of you do just that. What if there were no new antibiotic classes? And this is an interesting one because it's possible there aren't many new good antibiotic classes for monotherapy, because we've struggled in the last 20 years to find new ones. And the consequence of that thought would be that you would invest more in making novel macrolides, tetracyclines, lactam antibiotics, and so on. Actually, it turns out it's really hard for academics to do blue sky research in making novel compounds in these families. You just can't get money for it, and that doesn't feel right. It should probably be easier. And finally, what if there were no new targets? Because there's only half a dozen good targets for monotherapy antibiotics, okay? And we spent 20 years in the industry looking for new targets, and probably $100 billion, and didn't find any. So it's possible there are no good new targets for monotherapies. That's quite a bold statement. But if it's true, we should go back to the targets we know about and try to find new ways to inhibit them. And that's really hard to get money for as well. And those are a couple of things we probably have to change. Then we all talk about resistance. But is resistance the only issue? So last year, 10,000 people in North America died with MRSA. And yet all of those MRSA infections would be sensitive to linezolid and vancomycin, which were both available in North America. So why did those people die? And there's probably a variety of reasons, but possibly because the exposure at the site of infection wasn't right. And so personalized pharmacology or pharmacogenetics is probably going to be part of the future, and we don't invest in that. And then finally, the meat of what I'm going to discuss is, well, what are the alternatives? So if we can't find new antibiotics, what does the future look like? Is there a pointer on this? Do we have it? Okay. So what happened was the red one there. Thank you. The Wellcome Trust and the Department of Health thought it would be a good idea to collect some evidence on what the alternatives to antibiotics might be. And so they initiated and sponsored a review and a report into the alternatives. It was an open tender, and groups were invited to tender with a proposal for how they do it and a budget to provide this review. And I was one of the people who applied to provide the tender. And then there was the standard interview process and shortlisting and another round of interviews. And eventually I managed to win a contract to provide the service. And what I'm going to share with you is a bit of the story about how we did that. So first of all, we had to identify the scope of the review, and we felt it was too big to try and get into viruses and into fungi, and we just focused on bacteria. So our review scope was to look at non-compound approaches that target bacteria, because those would all be very different to traditional antibiotics, which are by and large compounds. And we also wanted to look at approaches that would target the human host or the animal host in veterinary healthcare. It had to be a suitable treatment to treat systemic or invasive infection. It could be administered IV, orally, or by inhalation. It could be monotherapy combinations, could be a prophylactic approach. But we avoided any topical administrations. We didn't want to get caught up in honey, silver, all the various things you can put onto wounds. And the approach I took was, not, instead of just doing it independently, was to form a working group to consider and collectively provide a consensus review to inform policy. And what the Wellcome Trust wanted, and particularly the Department of Health wanted, was a piece of work that they could use to inform policy and make a difference in the future. Not just a paper, they wanted some real impact here. And I don't know how you go to somebody and promise that in a couple of years' time you'll deliver a paper, because you can't guarantee what the reviewers are going to do, right? But you sit there in the interview and you say, oh yes, yeah, absolutely get a very high value publication. So the sorts of things we were going to look at were things like antibodies, probiotics, lysins. These are enzymes found in bacteriophage that you could clone, express and use as a medicine. and then antimicrobial peptides. So the sorts of things that insects use to stop getting bacterial infections. Could they be used in humans? So I created a working group of 24 characters. About half I would call drug discovery generalists, worked on traditional antibiotic approaches throughout their careers in biotech, academia, and in pharma. About half worked on alternative to antibiotic approaches, again, from academia, biotech, and pharma. About half from industry, half from academia. We had some regulators there. We had very early preclinical people. We had some clinicians. And I invited John Rex to co-chair the group with me. John was from AstraZeneca, a clinical microbiologist, a chief medical officer, so provided a good balance. And also importantly, he was involved in the US presidential sort of approach to developing antibiotics and was the Trust's expert in residence on antimicrobials. So it was important to me that we collect all the information and then you've got somebody who's going to still be embedded with the project in the future with the Trust. It's not just a standalone, you know, you get that learned and ongoing information. So the review process, the group came up with we found about 19 alternative to antibiotic approaches. And we could only use information that was in the public domain, academic papers, websites, and so on. We didn't use any proprietary information. And we identified 19 approaches. We reviewed those and created a 50-page, very technical report, which we shared with the Wellcome Trust and the Department of Health, and a few others. And then we had a meeting at the Wellcome Trust. It was a long day where we thrashed all those out. And then there was a subsequent email debate throughout the group, which led to the maturation and agreement of the technical report. And then the transformation of the report into a Lancet ID manuscript by a subcommittee of the group, but largely by John Rex and myself. After some interesting reviews, because we had everybody involved in the process, the major challenge that the Lancet had was finding anybody who could review it, because everybody was conflicted. But in the end, we managed to get around that. And it was eventually accepted and published online in January and in print in February. So we could at least tick the box that we'd managed to bring things together to a head and get it reviewed and published. So we looked at the alternatives in a number of different ways. We considered the feasibility preclinically. We considered the development issues that might face the projects, whether it was feasible to do informative clinical studies in the round. And then I was quite bad, actually. I asked this group of people to do something that particularly academics won't do, but industry people don't have a problem with, which was to rank them, okay? And I didn't give them the opportunity to rank things equally, right? They had to do proper ranking. And then I could take all the ranking of those projects and try to work out what the collective view of the group was. And I also mixed it up a bit because I put some traditional approaches to new antibiotics in with the alternatives, and then asked the group to rank everything. And this is the order. So basically the top ranked one, which for some reason, and I got my son, a teenager, to analyze this, because we did it all online, and he seemed to know how to do this. And for some reason, the best one's on the bottom. I never worked out why, but anyway. So the group thought that investing in tetracyclines, macrolides, all those, finding novel compounds on known scaffolds, is probably the best bet. Then we had alternative chemical means to hit the targets we know work clinically, so different ways to inhibit cell division, cell wall biosynthesis. Then we had next generation beta-lactams. So it's the same activity, but it's so important it came out on its own, right? Lactams are the best thing. We've got presentations over here. You know, we can keep them alive for a lot longer if we put the effort in. Then combinations of single gene targeting agents. So the last 20 years, we found a few compounds which work, but they're not good enough to be monotherapies because resistance develops. So maybe in the future, you could take two okay therapies, but not good enough for a monotherapy, put them together and make a new therapy. That might be possible. And then we started to get into the ones which are starred, which are the alternatives: antimicrobial peptides, vaccines, bacteriophage, phage lysins, and so on. And as it turned out, vaccines is a huge area, actually. And what we should have done was look at alternative approaches which are non-vaccines, because it's just massive. So I'm going to ignore vaccines for the time being and focus a bit more on the others. But what was interesting was you took this group of people, and some of them were industry people, didn't believe in alternatives. Some of them were dyed-in-the-wool alternative people. And we came up with a ranking which had about half alternative, half traditional approaches as the top half of the 19 that we identified. So alternatives to antibiotics, they're kind of a new technology. I was going to walk around, but they kept taking pictures, so I'll stand still. It's a new technology, okay? And like any new technology, it starts off, not many people use it in the market, it's innovative, it's led by visionaries, and then eventually it becomes a mature technology, it's widely adopted. You know, and this could be a mobile phone thing, you know, not many people use them and then everybody's got one. So the key question is, where are alternatives to antibiotics in this terms of maturity? So when you talk to an academic in this space, they say, well, I've discovered one, it has some efficacy in an animal model, I patented it because I wanted to publish it, it's almost a therapy, right? So it's mature, right? Why aren't there drugs? And then you talk to the people who've been doing drug discovery for a long time, and they would say, well, they're unproven, we don't understand the risks, it needs a much wider evidence base. We did a wide literature review. We only found 6 pharmacology studies for alternatives and 2 safety studies for alternative approaches in the literature. Now, you pick one of the drugs over there, one of the drug combinations, and you'll find many, probably 10 times more than that in the literature on one combination, not across all 19 approaches. We just don't know anything about how these things work, how to use them. So it's pretty immature in that sense. Commercially unattractive, and clinical trials that have been done have all been negative to date. So it's not that attractive, okay? So how do you bring all that together? And we know to bring it together and inform policy, there's a few things you've got to know. You've got to know how long it's going to take, how much it's going to cost, and what the risks are, okay? And so the way to do that is to do a portfolio analysis. And basically, we project planned all of the different versions of the 19 approaches that we could identify. So we created a great big Gantt chart and a massive great big spreadsheet with numbers on it. And we used widely agreed data to do that. So you can kind of predict that it's going to take about 5 years in research, a year in phase 1, a year in phase 2, 3 years in phase 3, a year to register to get a drug onto market. Now, these are pretty fast numbers because this is industry standard, which includes Me Too, different salt forms. You know, it's not widely, these aren't truly innovative projects, but they're reasonable numbers to start using. We know the stages of probability of success through each of these, and that gives you a cumulative probability of success. So when you start this process, you've got a 3% chance of success. And we know the costs, so we could budget, okay? So we created this understanding of the portfolio of alternative approaches based on these numbers. And then we could analyze the different styles of approaches and have a sense of the likelihood of success, how long it would take, and how much it would cost. I'll take you through this. Sorry, and then I'll take you through this one in a bit of detail, and then you'll zip through the others because you'll understand it. So this is phage lysins. So these are enzymes that you can inject into somebody's bloodstream, and they can completely degrade the bacterial cell wall, and the bacteria die. And in a test tube, you can inject a lysin into a culture of Staph aureus, and it clears in front of your eyes. It's in seconds. They're amazing things, okay? Now, there are 2 companies that lead this. Intron in, I think it's Korea, Contrafect in the US. At the time we did the study, they were kind of phase 1 ongoing, phase 1 ready, so early in phase 1. And if you're at that stage, they probably have if it all works, you could have a product by 2022. With 2 projects at this stage, that pipeline has a 26% chance of getting to market by 2025. So we can start to build in the metrics there. Now, you could say, if we wanted to believe in license and really test them and find out whether that is a new therapy for the future, just having 2 projects with a 26% chance of success isn't enough. You need more projects. So then what we did was, well, what would it cost to bolster a pipeline of an approach? And so you can say, well, if we want to do a phase 2 proof of concept study, one proof of concept study, you need 2 phase 1 projects to get there. You probably need 9 preclinical projects to get there. And that cost is £135 million. You're going to spend most of that, £112 million, in the first 5 years and put that at risk. But it gives you a sense of the budget. And it's important, if you want to influence policies, to let people know what these slugs of money look like. Okay? So if we wanted to invest in license and have a greater chance of finding out whether they work at phase 2, you'd probably have to inject about 135 million and over 100 million in the next 5 years. Okay? Does it feel likely anybody's going to do that? Probably doesn't, because money's a bit tight. Okay? So antibodies. Now, this is a much richer pipeline. These are all being trialed to prevent disease, not to treat it. We've got Merck and MedImmune, big companies. We've got biotechs, Aridis in, different forms of antibodies for C. diff, Staph aureus, and Pseudomonas aeruginosa. And they've all got a little pipeline. And at the time, we figured they could get market authorization in 2017. They actually managed and got market authorization in the last quarter of 2016. So you could probably buy and use this product now. This is a rich pipeline. There's over 100% chance of one of those reaching the market, and in fact, the most advanced one already has. So you wouldn't have to invest anymore in this pipeline. People obviously see it as attractive enough to do so themselves. And since then, we did this study. Aridis have released information showing that their version of these monoclonals may actually have therapeutic use. And actually, you don't need very much. You don't have to dose very much, and it still works in good animal models. So that's quite hopeful. So antibodies currently being developed to prevent infection. They may find use with, or potentially some of them could replace antibiotics. But bear in mind, the pipeline for almost all of these alternatives focuses only on 3 species: C. diff, Staph aureus, and Pseudomonas aeruginosa. And people have infections with more species than that. And almost nothing's being done for any other species in the pipeline. Okay. So antimicrobial peptides. There are tens of thousands of papers and literature that says antimicrobial peptides are the way to treat people in the future. Okay. So taking peptides from worms, peptides from insects, using them in humans, making mutant peptides, trying that. Okay. The most advanced project at the time was from a company called Polyphore. They had a Pseudomonas aeruginosa specific peptide, really good. All of the data looks fantastic. All of the preclinical models, efficacy, resistance frequency, potency, safety, very good. So good, in fact, they partnered it to Roche. Roche picked it up, pushed it hard. Since this paper, handed it back to Polyphore, not because it doesn't work, because they could see no route through regulation for a species-specific product at the time. So they handed it back. And it's very interesting now that Polyphore are working with the FDA very closely on trying to develop new paradigms to prove that these things are safe and efficacious. Because trying to deal with an agent that treats one species of bacteria is really hard to get the numbers of patients. You always have to treat these patients with a second antibiotic to cover other potential infections. You get confounding influences of that. And finding a safe regulatory path is the biggest challenge. For that product, it is an efficacy. Now, these other products are very early or effectively stalled. So with Roche handing that back to Polyphore and recognizing that its clinical development has stalled, then the chances of having an antimicrobial peptide went from 52% to down to less than 20%. So the chances are that's not going to happen unless regulatory environment changes. This is the product that Polyphore developed. It was created from or inspired by peptides. It's now a chemically synthesized peptide mimetic And I must admit, I made the case that it should come out of the review because it's a compound. But those obviously interested in peptides wanted to keep it in. But the real issue is that regulatory path. And when that's solved, that might open up the field to other styles of very specific products as well.